very good morning to all attendees today. Welcome to the next episode of the Breakfast at UMHealth webinar. We have a total of three interesting topics for the sharing session today, which will be presented by our FOM researchers. I'm Bavani Arumugam, moderator for the first two presentations of today's webinar. The session will be moderated by Professor Dr. Feng Xin Yi in the last session. Our first speaker today is Associate Professor Dr. Puah Suat, a senior lecturer from the Department of Biomedical Science. Let me introduce Dr. Puah briefly. Dr. Puah Suat received a PhD from UM in 2014. Dr. Puah's research interests include bacterial pathogenesis and investigating the function of virulence determinants with reverse genetic approaches. Dr. Puah has won many presentation awards at conferences and some prestigious awards such as the Young Investigator Award 2020, awarded by the Institute Marriott and the Malaysian Society of Infectious Diseases and Chemotherapy, as well as the APAC Australia Women in Research Fellowship. Dr. Puah is also holding several editorial positions such as section editor for the journal Gene Microbiology, associate editor for Gene Reports, and Asia Pacific Journal of Molecular Biology and Biotechnology. Today, Dr. Puah will be sharing some interesting updates on the insights into the current status of amoxicillin-based regimens for Helicobacter pylori eradication in Malaysia. With this, I would like to welcome Dr. Puah to present her findings. Dr. Puah, over to you.
Okay, thank you to Dr. Bavani. Thank you to Dr. Bavani. Okay, good morning to everyone. So I would like to share some information regarding genetic variations of penicillin binding protein 1A, which contribute to the amoxicillin treatment for Helicobacter pylori. Helicobacter pylori is a spiral-shaped bacterium which able to colonize human gastric lining and cause GIT diseases such as gastric ulcer and cancer. And H. pylori is one of the most common chronic bacterial infections worldwide. Two-thirds of the world population is infected. In Malaysia, a systematic review reported the prevalence of H. pylori infection is about 28%. And the strategy to eradicate Helicobacter pylori is recommended to follow Maastricht Foren's consensus report, which using a first-line standard therapy consisted of a combination of two antibiotics with a proton pump inhibitor. And amoxicillin is always paired with either clarithromycin or metronidazole. And in Malaysia, amoxicillin is used for the first-line therapy and second-line therapy. Let's look into some background of the antibiotic resistance in Helicobacter pylori from Malaysia. Based on the 11 publications on the primary antibiotic resistance, clarithromycin resistance has been studied for many years. But from the 1994 until 1999, there is a 0% of the resistance rate. But however, an increasing of the CLA resistance was shown in the 2014, 6.8%, and up to the nearly 15% in 2020. Which is reported by our group as well. So overall, the efficacy of clarithromycin has declined due to rising of the antibiotic resistance in H. pylori. Although Malaysia still are under a low CLA resistant region, however, our study showed an alarming result. So for the amoxicillin, unlike the clarithromycin, the resistance rate is consistently low. As you can see here, the report from 2011 until 2019, it shows 0%. But however, 2 years ago, there's a report from Malaysia reported 3.9% of the secondary resistance to the amoxicillin. How about in the World War? In the World War, actually the second antibiotic resistance to the amoxicillin is low as well, except from the Eastern Mediterranean regions, about 10%. And that is not recorded for the South Asia. This is the data up to the 2018. So in Malaysia, the amoxicillin remains is a key component for several H. pylori treatment. Amoxicillin is one of the first the better lactam first using to treat the H. pylori. So the 14 days standard triple therapy, which consisted of the proton pump inhibitor and two antibiotic, is a preferred choice of the H. pylori eradications. And another, there is high dose dual therapy, which consists of PPI and amoxicillin. Both actually are STT and HDDT show a high eradication rate. And the advantage using a HDDT is that it can eliminate the problem of the erythromycin resistance. So monitoring the background of the amoxicillin is very important to ensure the treatment success rate. And generally, the amoxicillin resistance occur due to the variants present in the penicillin-binding protein 1A, especially in the motif region, such as ST3K, TKTG. And actually, so far, there are 10 reported variants which found in the PBP1A and confirm their roles to contribute to the reduced susceptibility of the amoxicillin. Therefore, in our study, we would like to investigate the background of the PBP1A related to the amoxicillin, because we hypothesize that the high eradication rate in Malaysia, it could be due to the zero or low primary resistance rate of amoxicillin. So in our study, we collected 50 gastro biopsy samples from the treatment-naive patients. We bypass the cultivation step, which might take about 3 to 5 days. So we direct extract the DNA using a biopsy sample with a kit. And later to amplify the full length of a PBP1A, the size is about 2 kb. So we designed 2 sets of primer, and the purified PCR product were sent for the sequencing. And after that, we obtain the data and we align mapping to the ATCC strain 26695 using a bioedit software, and we translate to the amino acid and detect for any variants. Okay, so actually for the amoxicillin, amino acid variants in the PBP1A, it can be classified into two. First, there is a non-variance which have been reported in the literature review. So actually we refer as a reported variance, but for those no reports in the public domain and the new, so we classify as a novel. For the reported variants, we can subdivide into two groups, is that their function have been confirmed using a transformation assay. So this group of the variant, we are refer them as a validated variants, and another group that the function remains to be confirmed, and this group of the variants we refer as a reported variants. Usually they are detected using an e-test. And surprisingly, in our study, we found 21 amino acid variants, but however, they are consist of 9 reported variants, 12 novel variants, and none of the any validated variants with the confirmed function have been detected in our study. But this is somehow aligned to the previous surveillance data where the primary resistance rate of the amoxicillin is low. And actually, this also aligned to the treatment outcome involving amoxicillin in our study cohort. We're able to get the treatment outcome data for the 45 patients, and actually it showed a good eradication rate, about 80% of the success. And actually our finding also explain or support the superior of the HDDT, whereby H. pylori patients naive to treatments is able to be treated successfully using a amoxicillin-based treatment. But however, the resistant landscape for amoxicillin in Malaysia may be shifting. 2 years ago, the Sukri from Malaysia reported the first report about secondary amoxicillin resistant. And these two cases actually emergent might be due to the previous susceptible H. pylori strains developed resistance, because there is no report about primary amoxicillin resistant in Malaysia. And surprisingly, they also not detected any validated variants in these two secondary amoxicillin resistant strain, which is similar to our finding. And besides that, our result also found three amino acid variants which detected within or nearby the prohibitive motif. Okay, that is a K403X, S405I, E406K. And for the substitution E406K, or A, which have a conflicting data, because in the Cambodia, it present in the both amoxicillin sensitive and resistant strains. But in Japan, they only present in the amoxicillin sensitive strain. But for China, this substitution only present in the amoxicillin resistant strain. But for Vietnam, this substitution present in the sensitive and resistant strains. So far there is not defined answer can answer whether this substitution will play a role for the amoxicillin resistant. So actually more data using a more larger sample size, and the experimental using a transformation should be carried out to determine their functions. And we are also curious about the PBP1A sequences from the Malaysia. So that's why we use our 50 strain, plus we extract the sequence from the gene bank, 23 amoxicillin sensitive strains, which from the different country, Australia, Japan, Russia, USA, and so on. And another 20 strains from the amoxicillin resistance, okay, from Iran, okay. And then we construct the phylogenetic tree to see their genetic relationship using a meta software, and we visualize using an iTope. So from the phylogenetic tree here, there are several distinct clusters can be observed. But when we look at the Malaysian sample, which color in the gray, so that is from a unique group which distinguished from those other country. And we zoom into the Malaysian sample is that there is two major clades can be observed. The first one, they are clustered near to the amoxicillin sensitive strains ATCC 26695. And another two groups that is classified into two different clusters. So overall, we can say that actually the PBP1A sequence in Malaysia is considered unique and actually different from the from other country. And actually, and overall, actually, the molecular analysis of a PBP1A supports the therapeutic superiority of amoxicillin-based treatments. Antibiotic resistance will affect the therapy outcome. Therefore, it is very important to continue monitoring the amoxicillin resistant background in the H. pylori from our country, and to ensure actually the treatment's success rate is high. So now, actually, I would like to thank you to all the participants and our mentor, POFGO, and the funder, IRRG. And these are references for this study. And actually, our study have been just published in the Journal of Medical Microbiology. And if, let's say, you would like to access more information, you can go to access this article. And thank you, everyone.
Okay, thank you very much, Dr. Puah. So attendees, you're welcome to post your questions. At the moment, probably I'll have one question. Somewhere along your presentation, you mentioned that the amoxicillin resistant pattern is like shifting among Malaysians. So what does that indicate actually? What is the impact, or like what should be the further research move forward regarding this statement?
Okay, because the shift is that because it have many factor will be caused to the shifting. But what we can say is because bacteria and human, they will co-evolve. So actually, when we treat using a similar antibiotic, so it might be what to say, it might be immune. That's why that's why actually the case is very surprising is that we didn't see any primary resistance of the amoxicillin. But until there is a single report so far reported there is a secondary resistance occur. So that's why actually there is a lot uncertainty we haven't investigated, because not only PBP1A will cause the shifting of the amoxicillin, but actually there is another gene should be we need to expand the panel of the gene to monitor about this issue. Okay.
I see. Okay. Just a follow-up. So the PBP1, the unique panel, right, that you have presented in your last few
Yep.
slides about the unique property compared to other regions. So what does that indicates actually? So how does how does this propagates to the next level of the research?
Okay, the next level, actually, I would say, because PBP1A is the most common study for amoxicillin. Actually, usually people will focus on CLA, clarithromycin. Okay, so actually, there is compared to the clarithromycin, the study for the amoxicillin is less. So actually, I would say that there is certain like genetic variants we found nearby the conserved motif, which is worth to continue study to using a transformation to construct the mutant and the wild type, and to study about what is the level of the MIC that can be occur. So from there, actually, we can go back to tally about the dosage, and we can do something like adjustment. Yeah.
I see. Okay, okay.
Yep.
Understood. So thank you very much, Dr. Puah, for your sharing. And attendees are welcome to post your questions, and Dr. Puah may address your questions in the chat box or the Q&A box. Thank you very much. So